Hi everyone, welcome to my talk. First, I would like to thank SCIEX team for inviting me to present my work in this scientific series. I'm Pradeep Dewapriya, a postdoc research fellow working at the University of Queensland. I'm based at the Queensland Alliance of Environmental Health Science, generally known as QUES, which is located at the UQ Darton Park campus in Brisbane. QUES is a specialized institute at UQ dedicated to environmental health-related research activities. My work at QUES focuses on developing mass spectrometry-based analytical approaches to monitor human exposure to hazardous environmental contaminants. First of all, I would like to acknowledge the country where we meet today and to research. The University of Queensland acknowledge the traditional owners and their custodianship of the lands on which we meet. We pay our respect to their ancestors and their descendants who continue cultural and spiritual connection to the country. We recognize their valuable contribution to Australian and global society. This is the outline of my talk. I will briefly introduce you to PFAS, the forever chemical which we are struggling to understand and manage their effect on ecological and human health. And then I'll briefly talk about non-target analysis and its importance in PFAS exposure monitoring. And our non-target workflow and the work we did with workflow optimization, some of the results and future outlook. I think you have heard about some PFAS stories, maybe PFAS contamination and their health effects. But for those who are not familiar with PFAS, I'll give you a very brief introduction to PFAS, and this will be a brief introduction to PFAS and their sources. PFAS are a group of man-made compounds originally synthesized for non-stick and waterproofing coatings, but after realizing their performance, since late 1940, PFAS has been used in various industrial and consumer products due to their unique characteristics such as excellent surfactant capabilities, electrical and thermal insulation properties, water, oil, and date repellent properties. Here I'm showing you a typical PFAS structure. Most probably we can detect in all our bloods. This carbon-fluorine bond make these chemicals extremely persistent in the environment. Hence, these chemicals are generally known as forever chemicals. The properties that made PFAS an industrial success also contributed to PFAS extreme environmental persistence. Now PFAS has become a global environmental issue. In general, PFAS is highly mobile. PFAS can quickly move into environment from various PFAS containing sources and circulate through several pathways. Here I'm showing you some of the known PFAS sources that contribute to PFAS environmental contaminants. So PFAS move to environment through from these sources. Now we can detect PFAS in almost all environmental matrices from down under to polar ice regions in Arctic circles. Some studies have showed that PFAS in at least 97% of US citizens' blood. Are PFAS toxic? Yes, they are. This group of chemicals are associated with various health-related issues, including cancer, increased cholesterol levels, and immune suppressions. But unfortunately, existing studies are not thorough enough to give a definitive answer about the toxicity. More toxicological research is needed to confirm the direct link of PFAS to human health. Okay, what are the sources that contribute to PFAS human exposure? As I mentioned, these are the common sources that contribute to PFAS environmental contamination and human exposure. Among these sources, firefighting foam, generally known as AFFF, are the major source that contribute to PFAS environmental contamination and human exposure. PFAS containing AFFF became a global issue. Even in Australia, the use of AFFF largely contributed to PFAS contamination and human exposure. Here I'm showing you a timeline for PFAS use in Australia. Since 1970, both civilian and military authorities in Australia have used PFAS containing firefighting foams. After revealing the ecological and health impact of PFAS and the related compound, the use of PFAS containing AFFF gradually phased out. But still, this is the major source for PFAS contamination in Australia. This historical use of PFAS containing AFFF has contributed to over 800 contamination sites around the Australia. Humans, farm animal, and wildlife in Australia have been exposed to PFAS through these contamination sites. In addition to this major source, humans are exposed to PFAS through various household products that we use in day-to-day activities. In one of our recent review articles, we summarize 15 different household products that contain PFAS. Here I'm showing you the summary of this our review article. Some studies have reported extremely high level of PFAS in household consumer items such as household chemicals, textile, and food contact material. All these sources can contribute to human exposure. Because of that, now human exposure has become complex and be a human exposed to a very complex mixture of PFAS through all these the major sources and the household consumer products. As I mentioned, people exposed to a complex mixture of PFAS because PFAS is a complex group of chemicals with a huge structural diversity. The PFAS can be anionic, cationic, zwitterionic, or neutral compounds, come with various chain lengths and substitution. Depending on the definition, over 20 million PFAS compounds are currently listed in PubChem. All these indicate the complexity of the PFAS chemistry. HPLC is the most common laboratory-based analytical approach to monitor PFAS human exposure. Additionally, GCMS can be used for monitoring neutral and volatile PFAS. Depending on the pre-existing knowledge of chemicals, analytical approach can be categorized into three main categories: target analysis, suspect screening, and non-target screening. Target analysis detect and quantify the chemicals with known PFAS where reference standards are available. In general, target analysis provide with accurate quantitative information for the PFAS in a sample, which is that is very important for regulatory decisions. Suspect screening and non-target analysis are based on high-resolution mass spectrometry. These techniques are generally used to detect and quantify a broad range of compounds in a sample. As mentioned earlier, PFAS is a complex class of chemicals, and target analysis falls short of capturing the full PFAS profile because target analysis always focus on major PFAS in a sample with known compounds, leaving a significant portion of PFAS undetected. Several extractable organofluorine studies have further demonstrated that target analysis is not enough to capture the full spectrum of PFAS compound in a sample. Therefore, in past few years, non-target analysis and suspect screening employing high-resolution mass spectrometry have been developed as a solution to overcome this limitation. And the technique has been developed over the years now, and now non-target analysis and suspect screening is a popular and reliable detecting technique for a broader range of PFAS, including the chemicals that haven't been reported previously in samples. In our project, we aim to develop a non-target workflow to characterize PFAS in Australian population comprehensively. We aim to analyze PFAS in general population and occupationally exposed Australian using general population and firefighters' serum and blood samples. In this project, the key challenge is to develop a sensitive analytical workflow to detect and identify a broad range of PFAS in serum and blood samples. Optimizing analytical workflow is quite time-consuming and challenging. We had to overcome several challenges when we develop our workflow. These are the couple of challenges we faced, particularly the amount of samples that we had, both general population and firefighting serum and blood samples were quite precious to us and limited in quantity. Because of that, we couldn't use these samples for method optimization. We had to come up with another strategy. So we were looking for a set of samples that we can use for method optimization. As an alternative to our original sample set, we come up with another plan. We decided to use contaminated cow blood and serum samples to optimize our workflow. These samples were collected from one of the AFFF contaminated site near Brisbane. This contamination site is next to a military aviation center, and reports have shown that AFFF containing foams have been used in this center for long. These cows held next to the aviation center and have been exposed to highly contaminated groundwater for an extended period of time. So this was a kind of ideal sample for us. Previously, one of our team investigated the PFAS contamination in these samples using target analysis. Like many other PFAS monitoring campaign, this study only focused on a fraction of actual contamination, because in this study, only 10 PFAS were monitored due to the limited availability of reference standards at that time. However, the analysis reported samples with very high concentration of PFAS. We thought this is an ideal sample set to develop a workflow, because these animals have exposed to a complex mixture of PFAS from contaminated groundwater. So we optimize the sample preparation method using the cow blood and serum samples. And the sample preparation method used was the protein precipitation method. This is the most common extraction technique used for biomonitoring studies, but we slightly optimized the method to enhance the coverage of PFAS in our samples. This is the outline. This is the outline of our sample preparation method. In this work, we did not focus on individual animals because we aim to detect and identify as much as PFAS in these samples. So we decided to pool these samples. We mix equal volume of blood and serum samples from randomly selected cattle and extracted with acetonitrile. As a reference control sample, potentially clean animal blood and serum were also extracted using the same extraction protocol. Briefly, pooled samples were spiked with pseudo internal standard, which can be used as quality control and quality assurance standard in our data acquisition and data processing. And then we added acetonitrile to precipitate protein, and then the sample was sonicated for 30 minutes and centrifuge for 15 minutes using ultra high speed centrifuge to obtain supernatant. The pellet was discarded, and the remaining supernatant was dried under nitrogen and resuspended in 60% acetonitrile water mixture, and spiked with recovery standard, again to make sure that our extraction protocol is good enough to extract most of the PFAS in these samples. And then we developed we optimized the chromatographic method. For chromatographic optimization, we optimized the HPLC method to separate a broader range of PFAS. For that, we use C18 reverse phase column from waters. And using this column and chromatographic condition, we managed to optimize a method that can separate a broader range of PFAS. To improve our data acquisition and data processing capabilities, we injected all the samples in triplicate. These are the basic LC parameters we used for our method. And then we optimized the mass spectrometry method. For mass spectrometry analysis, we use SCIEX X500R QTOF system. For initial data acquisition, we use data independent SWOT analysis with 12 spot windows. For this optimization, we only focus on negative ionization, and both TOF-MS and MS-MS data was recorded for the pre-selected M over C range. And these are the other basic parameters that we use for the mass spectrometry method. Then move to the data processing and optimize the data processing workflow. In general, data processing is the most challenging part of non-target analysis. For data processing, again, we use a SCIEX OS analytics module. As I mentioned, data processing is a challenging and time-consuming part of the non-target analysis. So it took a fair bit of time to optimize the workflow. This is the data processing workflow that we optimize and use for analyzing contaminated cow blood and serum samples. In the next part of my talk, I will briefly talk about the different steps in my data processing workflow. Typically, blood and serum are quite complicated matrices. Here I'm showing you a typical total ion chromatogram of serum and blood samples. As I mentioned, these are complex matrices and very hard to detect and identify PFAS features from these samples. The challenge is not to lose low abundant features in these samples while reducing the number of noises we are extracting in feature detection. So we carefully optimize our feature detection, and for feature detection, we use SCIEX OS analytics module. As I mentioned, these are the basic feature finding parameters. Rest of the parameters were SCIEX default settings. Both blood and serum gave a massive list of features. We make sure that our extraction and our feature finding are quite good enough. We use SPIKE internal and recovery standard to confirm the quality of our extraction method and feature detection. All the samples gave us acceptable recoveries for standards. We were happy with the extraction method and feature detections. And then we moved to feature reduction in this workflow. As I mentioned, we ended up with a massive list of features because both serum and blood are quite complicated matrices. So we had to apply a series of filtering to reduce the number of features in our feature list. First, the frequency filter. Any features with relative standard deviation higher than 20% was discarded, and any feature coming from the blank was subtracted. In general, any feature with 10 times higher than blank were kept in our feature list, and rest of the things were discarded. And then the blank subtracted feature list was filtered through Kendrick mass defect filtering. For those who are not familiar with Kendrick mass defect filtering, in general, PFAS tend to have a negative mass defect due to replacement of hydrogen atoms by fluorine. So this replacement gives a negative mass defect to fluorinated compound. So KMD is a very useful filter to prioritize fluorinated compound in complex feature list. However, the KMD alone was not sufficient to reduce the number of features in our feature list. So we had to find another feature reduction possibilities. This is where our reference control sample supported us in reducing the number of features. We compared contaminated samples with reference control samples using SCIEX marker view software. Here I'm showing you the PCA analysis data for the contaminated and reference control samples. PCA analysis clearly shows us separate clusters for contaminated and control samples. Further, we performed a t-test using the marker view software tool to identify the features with high probability of detection only in the contaminated samples. Here I'm showing you the PFAS profile. PFAS was detected in high concentration in blood pool 5 and 6, but not in the pool 9 and 10, which are our reference control. For prioritization, features with p-value lower than 0.05 and log-fold change value higher than 0.9 were selected for further processing. This method was the most effective strategy for feature prioritization. Here I'm showing you a volcano plot to show you the t-test results. So as I mentioned, we selected this region of the volcano plot, and this was the best feature reduction strategy we found, and then that gives us a list of features that we can work on. But all the selected features in here were manually evaluated for the quality of the MS and MS data, and we examined the MS data to identify the PFAS specific fragments in these features. Based on the manual filtering step, finally we managed to prioritize 64 features for feature identification. For feature identification, a combination of library search, suspect screening, and non-target identification were performed. This is the library search that we performed. Library search were carried out with an in-house fluorochemical library integrated into the SCIEX OS software. This library consists of over 400 PFAS spectral data acquired in multiple collision energies. This library search were filtered through quality control criteria, and through this strict quality control process, we identified 12 PFAS, and all these 12 compounds were confirmed to level 1 identification, which means all these 12 compounds were confirmed with reference standards. All the identifications level I am going to talk about in these identifications are based on the recently introduced identification confidence scale. These are the different identification levels. In this scale, level 1A represent the confirmed PFAS with a reference standard, while level 5 here represent the mass of interest. Then we move to suspect screening. Suspect screening was carried out with a comprehensive list of PFAS specifically developed for AFFF chemistry. This suspect list was developed by the Chris Higgins lab at Colorado School of Mine. In addition to that, we use the NIST PFAS list, which contain over 3,900 unique PFAS formulas. For suspect screening, we use SCIEX OS quantitation and target identification workflow with the basic analytical parameters. Again, the suspect screening was carried out with rule-based filtering. These are the basic quality control parameters we applied to filter the suspect hits. Based on these quality criteria, 21 suspects, including PFAS, that were identified in library search, were identified. But manual quality control for these suspect hits were very important to avoid false positive and misidentification. So I will show you one of the example in our case. In our feature list, in the suspect screening, we found M/C value of 460.93, eluting at 5.09. And this feature gave a library hit as a perfluoroethyl cyclohexane sulfonate. However, in PFAS suspect list, there are several structural isomers that match to this monoisotopic match. So this is our SWOT data for this feature, and this SWOT data was not enough to confirm the structure. So we had to reanalyze the data with data-dependent acquisition mode to obtain a clear MS data. These are the data-dependent the data we acquired. With this data-dependent acquisition data, we confirm that the feature we detected in our sample was not the perfluoroethyl cyclohexane sulfonate. Actually, it's the unsaturated PFAS. So this is a good example to highlight the importance of careful analysis of MS data in suspect screening. Matching a compound to a monoisotopic mass is not enough to confirm the identification of a compound. So this is the full list of compounds identified through suspect screening. Particularly in our suspect screening, we identified many substituted PFAS highlighted here in red. These are not reported in our previous target analysis I mentioned at the beginning. Rest of the features were analyzed with non-target feature identification because those features did not give us any library match or suspect match. So this is a KMD plot prepared from the 64 prioritized features. In general, KMD plots help us to visualize the homolog series in samples. These are the homolog series we found in our sample. All the shapes in this plot represent a feature, and homolog series detected have highlighted in different colors. Through library search, suspect screening, and non-target analysis exercise, we managed to confirm 3 new PFASs in these samples and many non-compound. Interestingly, we saw a quite different PFAS profile in blood and serum sample. Particularly, we detected many short-chain sulfonamide and sulfonate in blood sample compared to serum samples. These are the MS spectra of the new PFAS we identified. Actually, this is not a very novel compound. This compound was first detected as from contaminated groundwater as a biotransformation intermediate. But for the first time, we detected this compound in biomonitoring samples, and this compound is completely new to PFAS chemistry. The structures of all these compounds were confirmed carefully with MS data analysis and diagnostic fragment identification. This compound class was prominently detected in blood samples, and this is the second new class we identified. This class was prominently detected in serum samples. Again, the structure of these two compounds were also confirmed by careful analysis of diagnostic fragments. This class is completely new to PFAS chemistry and haven't been reported in any environmental matrices before. This is the full list of compounds we identified from the samples with different identification confidence level. With our workflow, we managed to prioritize 64 features, including 27 known compounds and 3 novel compounds, as I mentioned. 34 features remain undetected due to the limited MS information to draw a structural conclusion. Okay, what we learned during our workflow optimization, we learned a couple of lessons from our optimization. Firstly, we identified 3 new compounds from blood and serum samples that are generally considered precursors. These compounds in general readily transform into stable non-PFASs, but detecting this compound in our cow blood and serum sample warrants further research on the fate of these so-called precursors. Secondly, the relative intensity of some of the compound detected in this study is greater than those normally monitored in biomonitoring studies. So these compounds couldn't be neglected anymore, I guess. And we noticed that there's a substantial difference between blood and serum profiles in cows. So it would be great to include both blood and serum extraction to capture true PFAS exposure profile in biomonitoring studies. Now we have a sensitive non-target analysis which can detect a broader range of PFAS compounds, including compounds that were not reported before. So now we are ready to analyze our original samples as we planned before to capture PFAS profile in Australian population. What's next? At QuAIS, we have collected serum and blood samples for many biomonitoring studies. We have samples from firefighters, communities knowingly exposed to PFAS, and the serum samples from general population from 2002 to today. So now we are going to analyze all these samples to characterize the PFAS exposure in Australia comprehensively. So this work is in progress. Currently, we are analyzing the data. So I hope in near future I can show you the results from this sample analysis. With that, I would like to conclude my talk and thank all who helped me to continue this work, especially my team at Quest. And especially, I would like to thank SCIEX team for inviting me to present our work in this PFAS Summit series. And finally, all you for your kind attention. Thank you.